Okay, I'm wondering if we could review creation. Yeah. 1, 2 motion. Does this stay the size there is? No, that's why I close expensive to buy. Yeah, then you know that really in their first the company on this cost is 0.
Yeah, I use it.
Just one of them. I guess he's sitting in the back. We're just waiting on 2, right? It's just Dr. Dennis and Dr. Thomas.
Next week.
Stable media.
Okay, so probably we can start. So it's really my pleasure to introduce the lead start in my life as a PI student in his junior. So at that time, I still do write students exam when they came to my life. I stopped doing that now because now I mostly take a freshman and a sophomore. So Denis actually didn't do very well, right? But it's okay. At least he knows how to make working buffers from stocking solutions, right? And know how to adjust the pH. And so anyway, after a semester, right? So I started with painting, and he finished. I guess it's one semester early, right?
Yeah.
And but I didn't know what to do, so I paid him as a technician. And later, he came to me. He said, "I want to apply to graduate school." It's kind of late in the season, so I asked him, "Have you taken GRE?" He said, "What is GRE?" I didn't make it up. So anyway, he took GRE the next week, and he did it very well. Although I never taken GRE, I know it's hard, and students in China have to prepare for years to take GRE. But anyway, he did well, and he started in 2010, and I think he struggled a little bit, and until his fourth year, right, he didn't have a publication, right, and although he has generated a lot of data, when he applied to F31 fellowship, so I was not very confident because there's no publication. But anyway, he got a F31, in the pre-doctoral fellowship from NIH. So that's why I encourage every student just give a try, right? And now he's ready to defend his dissertation. He has the fellowship He has first author publication in PLOS patent, and also a journal of virology paper and revision. He has many co-author papers. So he also has postdoctoral position. He only applied to one institution, one university for his postdoctoral position. He may explain why he only applied to one position. So anyway, so I'm going to stop here. Now it's your show.
Thanks. So I meant to ask you earlier today to keep the introduction brief, but I forgot. That's my fault for not. So I just want to start by thanking you all for coming to my dissertation defense. And the title of my talk today is Modulation of Kinase Signaling by Kaposi Sarcoma-Associated Herpesvirus. So this is a general outline of the talk. So I'll start with some basic background and introduction to get everyone on the same page. Then the bulk of the talk will consist of phosphoproteomic and transcriptomic analyses of KSHV-infected cells. Finally, the last section is sort of a side project that stemmed from this major project regarding a functional interaction between two KSHV proteins. So our lab studies KSHV, which is a herpes virus, and this is an electron micrograph of a typical herpes virus structure. In addition to the genome and capsid that all viruses possess, it also has an envelope layer studded with glycoproteins and this amorphous intermediate layer called the tegument, which is comprised mostly of proteins. And this tegument layer is the major focus of our lab for reasons I'll discuss later. So there are totally 8 human herpes viruses, and we're probably all infected by at least one of these, even if you don't know it. So most people are infected by EBV, which is the causative agent of mono, at a very young age because it's so ubiquitous in nature and also so efficient at maintaining persistent infection. And that's why you may have heard the phrase, herpes is for life. So what you might not have known is that 2 human herpes viruses can cause cancers. These are EBV and KSHV. So these are 2 of 7 total human oncoviruses, or viruses known to cause cancer in humans. And you're probably aware of the links between some of these and cancers, for example, HPV and cervical cancer, or hepatitis viruses and liver cancers. The virus we study is KSHV, and it's the cause of 3 human malignancies. These are Kaposi sarcoma, or KS, and 2 B-cell lymphomas. So these diseases are particularly prevalent and problematic in immunocompromised individuals, including AIDS patients and organ transplant recipients. KS was originally described in the late 1800s as the sort of rare and mild skin lesion, usually affecting elderly men, something that affected individuals would die with, but not of. But at the start of the AIDS epidemic, it was seen more and more frequently in previously healthy young men, and it took on a much more aggressive form, often spreading to the visceral organs, not just the skin. And this spurred a huge research effort to identify the causative agent, which was eventually shown to be a herpes virus in the early 90s and named KSHV. So KSHV is a large double-stranded DNA virus, and some of its complexity becomes apparent when looking at the KSHV genome. So it encodes about 90 genes, and importantly, many of these, the functions are still not well characterized. This diagram summarizes the typical herpes virus life cycle, so I'll summarize this briefly. Once the virus enters the cells, the tegument proteins are released into the cytoplasm. These are these little dots here. And the capsid and genome is delivered to the nucleus. At this point, the virus can enter one of 2 alternative life cycles, latency or lytic replication. During latency, only a handful of genes are expressed, and it allows the virus to escape evade immune detection and also replicate the viral genome undetected. During the lytic cycle, the full complement of viral genes is expressed in a temporally regulated cascade, leading to the production of new viral particles and ultimately lysis of the infected cell. So our lab mostly studies lytic replication, and we're interested in the functions of lytic proteins, more specifically tegument proteins, because as I mentioned, these are released immediately upon infection, so they can have roles relating to suppression of the host innate immune response, and they're important for establishing latency. But they can also have roles during the lytic cycle, for example, in processes such as facilitating efficient expression of viral lytic genes and also in the assembly process to make new viral particles or virions. So over the next few slides, I'll give some more specific background related to my project. So as I mentioned, we're interested in tegument proteins, and much of my work is centered around one KSHV tegument protein called open reading frame, or ORF45. And shown here is one of the major functions of 45, many of which have been discovered by our lab. And this just shows that ORF45 activates two cellular kinases, ERK and RSK. And this is evidenced here by increased phosphorylation of the RSK substrate S6 ribosomal protein in the presence of ORF45. Importantly, the binding to and activation of RSK can be abolished by a single point mutation, F66A. And I'll come back to this point mutant several times throughout the talk. In order to study the roles of ORF45 and RSK in the context of lytic replication, we required a model system. So this diagram shows a system that we used. We started from the SLK cells, which are endothelial lineage, and were derived from a biopsy of a Kaposi sarcoma patient. So to these cells was added a TET-on system, so that when the cells are treated with doxycycline, it induces expression of RTA. RTA stands for replication and transcription activator, and so this is the major lytic switch protein. Its expression is necessary and sufficient to initiate the full lytic cycle. If to these cells we add a bacterial artificial chromosome clone containing the entire infectious KSHV genome, and select to maintain a stable cell line, now these cells are latently infected, but they can be chemically induced with doxycycline to undergo the lytic cycle. Importantly, because the BAC can be manipulated and propagated in E. coli, we can make targeted mutations to any gene of interest and then study the effects on viral replication. So we did this with ORF45 by making the F66A point mutant as well as an ORF45 deletion mutant termed stop45. We also made the revertant mutants back to the wild type sequence to ensure that whatever phenotype we observed for these was due to the targeted mutation and not some off-target effect. We confirmed that only the intended mutations were made by RFLP and also Sanger sequencing, and transfected these backs into ISLK cells to make stable cell lines. So we induced lytic replication in these cells and collected media at different times post-induction. And what's shown here is a measure of viral genome copy, or basically virion production, at different times post-induction. What you can see is that compared to the wild type or revertant cells, the F66A mutant virus-infected cells is dramatically deficient in virion production by more than an order of magnitude, almost to the same extent as completely deleting ORF45. So this suggests that the activation of RSK by ORF45 is important for efficient virion production. Now, we also performed RT-PCR of all of the KSHV genes and Western blot of different viral proteins and found that viral late gene expression is compromised. So at this point, we know that viral late gene expression and virion production are dependent on ORF45 and RSK, but we didn't know the underlying mechanisms. So uncovering these mechanisms was one of the major goals of my dissertation. Based on our data and what we knew about the functions of RSK, we hypothesized that during the lytic cycle, ORF45 activation of RSK contributes to differential phosphorylation of several substrates, which would thereby modulate their activities. And in particular, we were thinking substrates related to transcriptional and translational regulation, which could explain the differences in viral gene expression, and that this was a mechanism to maximize viral particle production. So based on this hypothesis, then two specific aims. First, we wanted to identify and characterize the substrates of ORF45 activated RSK. And second, we wanted to elucidate the mechanisms by which ORF45 and RSK contribute to viral and cellular gene expression regulation. So shown here is the family of AGC kinases, which RSK is a member, or P90 RSK. That also includes AKT and S6 kinase. And here are the upstream kinases that converge on activation of these AGC kinases. As a reminder, ORF45 acts at this node here by binding to and activating ERK and the downstream RSK. So importantly, AGC kinases share a common substrate phosphorylation motif, which is a serine or a threonine preceded at the -3 and -5 position by an arginine. And the downstream effects of phosphorylation of these substrates are wide-ranging, affecting key biological processes. Just to note, in dark blue here are different KSHV genes, and in red are various kinase inhibitors used later in the study. So we first asked a very simple question. Are AGC kinase substrates, those phosphorylated at this motif, differentially phosphorylated in KSHV-infected cells? This would indicate an altered activity of one or more of these AGC kinases. And indeed, that's what we saw. So shown here is a Western blot of KSHV-infected cells, and this is a time course of lytic reactivation. The U here stands for uninduced, where you can consider latently infected, whereas the I is induced to undergo lytic replication. And especially at later time points, coinciding with expression of ORF45 and activation of RSK, we saw dramatically increased phosphorylation of proteins that contain this motif. However, in this system, it was impossible to tell which of these proteins were true substrates of RSK and which could also be phosphorylated by AKT or S6 kinase, since they're all activated during the lytic cycle and they all share this substrate phosphorylation motif. To get at this, we used our ORF45 back mutants. And just as a reminder here, the A66F is the revertant back to the wild type sequence. Importantly, we found that this increase that was induced by lytic replication is almost completely lost in either of the ORF45 mutants. And we did assess the activities of AKT and S6 kinase in these cells and found no significant difference between the revertant and the mutants, suggesting that this difference that we observed could be attributed to a difference in RSK activity. So because this effect was so clear, it led us to believe that we could identify the substrates of ORF45 activated RSK by mass spectrometry. So this just shows a flowchart of that experiment. We compared the A66F revertant to the F66A mutant at 2 days post-induction when we saw the biggest difference in their phosphorylation profile. Importantly, we included an immunoaffinity purification step to enrich for phospholipeptides that contained this RSK phosphorylation motif. So shown here is a table of the top hits from that mass spec analysis. And they're ordered by a fold change in phosphorylation. In other words, these are genes whose phosphorylation was reduced by F66A mutation, indicating that they may represent real substrates of ORF45 activated RSK. And while many of these are interesting and have potentially relevant functions to KSHV pathogenesis, for the purposes of today's talk, I'm going to focus solely on EIF4B, which is a translation initiation factor. And I'll talk later about the importance of this protein and its phosphorylation during the lytic cycle. So the total phospholipeptides can also be viewed as a scatter plot. Now on the x-axis is fold change in phosphorylation. So these outliers to the right represent many of the same genes shown in the previous table. If we filter out all of the phospholipeptides with at least a two-fold change between the revertant and the F66A mutant and arrange them by protein type or function, what becomes clear is that proteins in a vast variety of different types or functions are affected by ORF45 and RSK. But even more strikingly is that about a quarter of these most significant hits are transcriptional regulators. Now, we also performed gene ontology analysis on the same subset and found that terms such as nuclear localization and transcription were enriched, as were things we expected like phosphoprotein or host virus interaction. So we next wanted to further investigate the roles of RSK and its downstream substrates during the lytic cycle. And to do this, we made use of the CRISPR-Cas9 system. So a couple years ago, I probably would have had to explain this in great detail, but as Eric Lander wrote in his recent and hotly debated review article, if there are molecular biologists left who have not heard of CRISPR, I have not met them. So as you probably are aware, this is a system to induce a targeted cleavage, usually in genomic double-stranded DNA, although the nucleases are constantly being modified for other purposes, and every week there are new and exciting applications to biomedical research. At the time I was designing these experiments, the lentiCRISPR or lentiviral vector, which contains the CRISPR and Cas9, the CRISPR guide sequence and the Cas9 nuclease, was just being developed. So we used this system to knock out both RISC1 and RISC2, the two isoforms affected by ORF45 in KSHV infected cells. So as evidenced by Western blot, there's no detectable expression of RISC in these knockouts. And just to clarify here, number 1 and 15 represent two single cell clones, both of which have knocked out RISC1 and RISC2. So if we look at overall phosphorylation at this motif, you can also see that it's dramatically reduced in the knockouts compared to cells transduced with the empty lentiCRISPR control. Now we see the same sort of effect when we look at individual substrates, and I won't go into the validation and functions of many of these, but again, I'd like to draw your attention to EIF4B, whose phosphorylation induced by KSHV was almost completely abrogated by RISC knockout. The knockout also reduced viral late gene expression. So on the left here is RT-PCR. In the green boxes are early genes, and the red box is late genes. So you can see that the values of late genes are decreased, and you can see the same trend if we look by Western at proteins. Finally, the knockout reduced progeny virion production by about tenfold. So all of these phenotypes, the decreased phosphorylation, decrease in viral late gene expression, and the virion production were reminiscent of and consistent with the F66A phenotype. And they serve to confirm the importance of RISC during KSHV replication. Now, going back to this list of substrates, we also wanted to investigate the potential roles of these proteins during the lytic cycle. So we used a similar approach to systematically knock out each of them in KSHV infected cells and then assayed virion production at the latest time point. And what we found is that many of these knockouts were significantly deficient in virion production. Again, I'd like to call your attention to EI4B, which had one of the most dramatic effects that we observed. So we wanted to further characterize the functions of this protein with particular regard to translational regulation. So first, we confirmed that it is phosphorylated in an ORF45 and RISC-dependent manner. On the left, you can see that in the ORF45 mutant cells, its phosphorylation is decreased. And on the right side, we treated with a panel of kinase inhibitors, and only the RISC inhibitor was able to decrease EI4B phosphorylation that was induced by lytic replication. So in order to understand the next couple pieces of data, I think it's helpful, if not necessary, to have a general understanding of the process of translation initiation. So I'll try to summarize it briefly. I don't pretend to be an expert, and it's a complicated process, but for the purposes of my talk, you really only need to know two components. These are EIF4A and EIF4B. So 4A is the RNA helicase, which is responsible for unwinding secondary structures that often form in the 5' untranslated region, or UTR, upstream of the start codon. EIF4B interacts with both the RNA itself and EIF4A and enhances the RNA helicase activity of 4A. And it's been previously shown that phosphorylation of EIF4B at the site we identified is important for this function. Furthermore, a study published a couple years ago in Nature showed that EIF4A is important for the efficient translation of genes containing G quadruplex or G4 structures in their 5' UTR. So because of the known role of EIF4B in enhancing 4A activity, we thought that EIF4B may also play a role in this effect. So to test this, we used a reporter assay using a G quadruplex containing luciferase reporter in the presence or absence of ORF45. So what we can see is that ORF45 increases the luciferase activity of the G4 containing reporter, but not a control of equal GC content. And that this effect is dependent on EIF4B because the same increase is not observed in an EIF4B knockout cell line. Now we also have data to suggest that the translational regulation by ORF45 is important in the context of KSHV infected cells. So to understand this data, I'll need to describe the experiment a little bit. So what we did here is, using these ORF45 mutant cells, I induced lytic replication, and at 2 days post-induction, treated cells with cyclohexamide to stall translating ribosomes. I then harvested lysates and fractionated them and analyzed the RNAs on a sucrose gradient. So this is the typical pattern that you might expect to see, where RNAs are associated with monosomes and also polysomes in the heavier fractions. So on the right panel, on the y-axis here is a ratio of polysomal versus monosomal RNAs. And this is traditionally thought of as a measure of translation efficiency, because transcripts with multiple ribosomes translating the same transcript are being translated more efficiently than only a single ribosome. So you can consider this y-axis value as a measure of translation efficiency. On the x-axis are various genes, mostly KSHV genes, and they're ordered by the complexity of their 5' UTR. So complexity is sort of a vague term. More specifically, these are ordered by their minimum free energy. And what we can see is that genes with more complex, longer, structured 5' UTRs are less efficiently translated in the ORF45 mutant cells compared to the revertant. We performed similar experiments in EIF4B knockout cells and saw a similar effect. So this diagram summarizes the direct and indirect mechanisms by which ORF45, through activation of the cellular kinases ERK and RSK, regulates protein synthesis. But if you remember from the phosphoproteomic data set, I showed that many of those genes were transcriptional regulators, and yet I focused on translation. So the question remained, how are ORF45 and RSK involved in transcriptional regulation during the lytic cycle? To get at this, we performed RNA-seq of KSHV-infected cells. So the next few slides show the experimental design for this experiment. We included 12 samples, and the first comparison we wanted to make was between uninduced and induced revertant cells. So this would basically just give us an indication of the effect of lytic reactivation. Now, of course, we were also interested in the effect of ORF45 and RSK, which could be inferred by comparing these ORF45 mutant cells to the wild type or the revertant. And we performed this in biological duplicate. So we depleted ribosomal RNAs and created index libraries using kits from NEB, then ran these on the Illumina HiSeq and the College of Medicine. So this slide shows the overall differential gene expression as a consequence of lytic reactivation. This is comparing the induced to uninduced, just the wild type virus-infected cells. And what you can see is that, especially at later time points, there are several hundred, so this number is 500, genes that are upregulated as a consequence of KSHV lytic replication. And there are also some significantly downregulated genes. So this was not surprising, and so perhaps the more exciting and novel insights to be gained from this experiment was the effect of ORF45 and RSK. If we look at just the KSHV genes, we do see a significant difference in gene expression. So this is a heat map comparing the revertant to the F66A or the STOP45 mutant at either 24 or 48 hours post-induction. And this is every KSHV gene clustered by expression kinetics. So latent, immediate early genes, early, and late. So what you can see is that, especially at the later times, there's a significant difference in the expression of several late genes and also some early genes, while immediate early and latent genes are not appreciably affected. So this is consistent with RT-PCR results that we obtained from these same cells and confirms the role of ORF45 and RSK in the regulation of KSHV gene expression. As for cellular genes, we performed KEGG pathway analysis, and over the next few slides, I'll show you some of that data briefly. So we saw significant differences upon F66A mutation in components of the basal transcription factor machinery, apoptosis signaling, cell cycle regulation, and finally cytokines and cytokine receptors. Now this last one is particularly interesting because it's been known for several years that the increased production of cytokines and chemokines by infected cells plays a dominant role in the progression of KSHV-associated diseases. And I don't expect you to take anything specific away from this or the previous few slides. I mainly wanted to make the emphasis that ORF45 and RSK seem to affect diverse pathways that are important for KSHV pathogenesis. So to summarize this section, I've shown you that KSHV induces the phosphorylation of several putative RSK substrates. Phosphoproteomic analysis confirmed that many of these are phosphorylated in an ORF45 and RSK-dependent manner. We assess the consequence of knocking out either RSK or its downstream substrates on virion production. ORF45 and EF4B appear to be important for the efficient translation of genes with structured 5' UTRs, including G4 structures. And finally, RNA-seq analysis reveals diverse functional consequences of ORF45 and RSK on transcriptional regulation. So for future directions, we're excited about following up on many of these results, including the RNA-seq analyses. And as far as the translation side, we would like to perform ribosome profiling of KSHV-infected cells to get a more unbiased analysis of the effect on translation. So for this last section, I'm going to switch gears a little bit. And as I mentioned, this is a side project. And so this is something I've been working on sort of intermittently alongside this major project. Something we noticed from the mass spec analysis is that there was only one KSHV protein that had this RXRXX-ST motif that was differentially phosphorylated between the wild type and F66A mutant virus, and this was ORF36. So furthermore, we previously noticed that the level of ORF36 is decreased in the F66A mutant. So we wondered what's the relationship between ORF45, RSK, and 36. Taking a step back, what is ORF36? So ORF36 is a serine threonine protein kinase, and it's the only one encoded by KSHV. It's a member of the conserved herpesvirus protein kinases, or CHPKs, and the homologs are shown here for other related human herpesviruses. This figure illustrates the known functions of CHPKs across one or more of the 3 herpesvirus subfamilies, alpha, beta, and gamma. So some of the major conserved functions shown in red are phosphorylation of the lamins, which induces their disassembly and is important for newly formed nucleocapsids to get out of the nucleus and into the cytoplasm. It's also been shown to play a role in innate immunity by inhibiting the expression of interferon beta. So, but compared to its homologs in other human herpes viruses, there's relatively little known about KSHV or ORF36. There's maybe only a half dozen papers, each describing a single substrate. So because of this, we wanted to investigate the roles of this protein, and particularly the roles during the lytic cycle. But first, we wanted to just confirm its phosphorylation by ORF45 and RSK. So shown here is just a co-IP experiment. When we co-express ORF45 and flag tag 36 and immunoprecipitate 36, we can see increased phosphorylation of 36 at this RSK phosphorylation motif, so, suggesting that ORF45 and RSK are important for its phosphorylation. But we also found something that we didn't necessarily expect to see from this experiment, in that ORF45 stably interacts with ORF36. And also this complex appears to include RSK and ERK as well. Importantly, these phenotypes, the increased phosphorylation and the binding, appear to depend on ORF36 kinase activity, because a kinase dead or KD mutant is incapable of being phosphorylated or interacting with ORF45. So because of the interaction, we wanted to map the domains in order to learn more about this interaction. On the left panel here, we use GST pull downs to show that the N-terminal 83 amino acids are necessary and sufficient for the binding, because any mutants lacking these N-terminal 83 are incapable of binding to ORF45. On the right panel, we used a series of ORF45 internal deletion mutants and found that deletion of only one region, amino acids 90 to 115, was incapable of binding. So this absence of signal in these other lanes is marked by asterisks, is due to the epitope of one of the monoclonal antibodies that we use to detect ORF45. So we confirm these GST pull-down results with co-IP experiments. And on the left, you can see that, again, any mutants of ORF36 lacking the N-terminal 83 amino acids exhibit dramatically reduced recovery of ORF45 after immunoprecipitation. On the right panel, we used different ORF45 mutants. So this delta 90 to 115 that didn't bind in the GST pull-down, as well as 3 mutants of ORF45 that are incapable of activating RSK. Interestingly, we found that any of these mutants didn't bind to ORF36. So it suggests that not only this binding domain may be real, but any mutant that is deficient in RSK activation also doesn't bind. So it suggests RSK is important for the interaction. So this 90 to 115 region is interesting because 90% of the amino acids in this region are glutamic or aspartic acids. So it's a heavily acidic region of ORF45. Whereas ORF36 is a basic protein with the PI of around 9.4. Furthermore, the PI of this N-terminal 83 amino acids is 11.4, which is extremely basic. So based on this, we hypothesized that the optimal interaction between 36 and 45 might involve electrostatic interactions between negatively charged 45 and positively charged ORF36. To test this, we performed the GST pull-down assay, but this time included lambda phosphatase to dephosphorylate purified ORF45 protein in the presence or absence of phosphatase inhibitors. So what you can see is that the phosphatase-treated ORF45 doesn't bind as well to ORF36 as the untreated, whereas inclusion of the phosphatase inhibitors almost completely recovers the binding. So this suggests that the phosphorylation status and potentially the charge of ORF45 is important for the interaction with ORF36. Because of the effect of the ORF45 mutants in the previous slide, we also wondered what's the importance of RSK for their interaction. So to get at this, we made RSK knockout cells in 293Ts, and shown here just 2 single cell clones, and you can see that the protein is completely knocked out. And we performed a co-IP and found that in either of these knockouts, there's dramatically reduced recovery of ORF45 compared to co-IP from wild-type cells. So this suggests that the presence of RSK is important for the binding between ORF45 and 36. Denoted by this arrow, we also see decreased phosphorylation of ORF36 at the RSK phosphorylation motif, which we would expect. So going back to this slide from the introduction, we also wanted to understand the effect on ORF36 protein. So certainly this could be attributed to transcriptional or translational effects of ORF45, which I pointed out in the previous section, but the effect was also reminiscent of ORF45's effect on another KSHV tegument protein, ORF33. So this is work done by Joseph Gillen, a previous graduate student in the lab, and what he found is that ORF33, when expressed alone, is rapidly degraded after cyclohexamide treatment, which inhibits new protein synthesis. However, in the presence of ORF45 wild type, the ORF33 protein level is dramatically stabilized. So I performed a similar experiment and found that ORF36 also is degraded within 8 to 12 hours, but in the presence of ORF45, it has prolonged stabilization. So the effect seems to be very similar to what he found. Furthermore, the kinase dead mutant is dramatically less stable than the wild type. So it's almost completely degraded within a couple hours of cyclohexamide treatment. And this protein is not stabilized by ORF45. Finally, MG132 treatment suggested that the normal degradation of ORF36, either the wild type or kinase dead mutant, is dependent on the proteasome. Now we went on to find that the N-terminal 83 amino acids, the same region involved in the binding, seems to be important for this effect because any truncation mutants of ORF36 that possess these 83 amino acids are able to be stabilized by ORF45, However, mutants lacking the N-terminal 83 amino acids are not stabilized. So at this point, I decided to make mutants in the back, similar to what we did for ORF45. So I made the kinase-dead point mutant, as well as 2 deletion mutants, termed stop36 and delta star 36. So the stop is insertion of premature stop codons just after the start codon, and the delta star is the mutation of the start codon itself. So this was somewhat tricky due to the overlapping open reading frame 35. However, I was able to make minimal substitutions so as not to affect that amino acid sequence. So we first confirmed that in the deletion mutants, ORF36 protein is gone. And in the kinase-dead, the level is slightly lower. We think that this is probably due to the lower stability of the kinase-dead mutant. And we looked at several other KSHV genes. The top 4 here are all immediate early and early genes. So because ORF36 itself is a late protein not expressed until 2 days, we didn't expect for it to have much effect on immediate early or early gene expression. And that's what we see is no significant effect for these. However, for most of the late genes that we tested, including these bottom 4, which code for capsid and tegument proteins, we do see a significant reduction, especially in the kinase-dead mutants, compared to the wild type. Importantly, we confirmed the interaction between ORF45 and 36 in KSHV-infected cells. So this is shown here using IP with an anti-ORF45 antibody. We can detect ORF36 as well as this. However, upon F66A mutation or ORF36 kinase-dead mutation, we see decreased recovery. And this is consistent with the co-transfection results. We also confirmed the effect on stabilization in KSHV-infected cells. So what you can see here is that in the stop45, which doesn't express ORF45, ORF36 is degraded more rapidly after cyclohexamide treatment than the wild type. And also in the kinase-dead mutants, at the later time points, you can see the reduction of signal to a further extent than in the wild type. Because we saw decreased expression of several viral late genes encoding structural components of the virion, we thought that virion production might also be compromised. So indeed, this is what we saw. For the kinase-dead mutants, it's almost a 10-fold reduction, which is comparable to the F66A mutation or risk knockout. However, when we normalize these virions by viral genome copy number and compare their protein composition, there is basically no effect, no significant effect for the proteins we looked at. So suggesting that ORF36 is not important for the efficient packaging of new viral particles. The only difference is the inclusion of ORF36 itself. Obviously, it's not present in the deletion mutants. So because we detected ORF36 in the virions, we wondered, is this enzymatically active? So to test this, we performed in vitro kinase assays of purified lysed viral particles. So we just lysed the particles and added hot ATP and basically ran the entire reaction on the gel to see what proteins could be phosphorylated. And interestingly, there's this strong diffuse band at the exact expected size of ORF45 that's dependent on ORF36 because in the deletion or kinase-dead mutants, we see a significant decrease, suggesting that this could be a viral or virion substrate of ORF36. And there's also these upper and lower bands we detected as well. So current experiments by undergraduates in the lab are aimed at further confirming and actually identifying the specific virion components phosphorylated by ORF36. So this was interesting and exciting to us because not only did it suggest that ORF36 is enzymatically active in virions, but this could have implications for primary infection. To clarify, going back to this introductory figure, these little dots dissociating from the capsid represent tegument proteins. So it's been shown in other herpes viruses, namely EBV and herpes simplex virus, that the dissociation of various tegument components from the capsid relies on phosphorylation by their homologue of ORF36. So the CHPK of these other herpes viruses is important for inducing the dissociation of these tegument proteins. However, no one's looked at this for KSHV. Over the next couple slides, I'll show data to suggest an important role of ORF36 contained within the virion in establishing primary infection. So one of the advantages of the BAC system is that it possesses a GFP marker within the BAC that's constitutively expressed as long as cells are infected. So you can see here just from fluorescence micrographs, when we infect cells, you see the GFP signal. And if we perform FACS on these cells, what we notice is that here we're using the same input amount of starting virus. However, for the deletion mutants, there's a decrease in the percentage of GFP positive cells. This suggests that ORF36 is important for the efficient infection of these cells. And you can see that from the FACS and also just from looking at the fluorescence micrographs. And this figure shows for each MOI, or multiplicity of infection that we tested, we see the similar ratio of decrease upon ORF36 mutation or deletion. Compared to the wild type, there's at least a 2 to 3-fold defect, as much as a 5-fold decrease in infectivity. So to summarize this section, I've shown you that ORF36, 45, and RSK exist in a stable complex. We mapped the interacting domains. We found that ORF45 enhances 36 stability, and I didn't show this data due to lack of time, but it also enhances the in vitro kinase activity of ORF36. And in the context of KSHV infected cells, we confirm the interaction and the stabilization effect, as well as finding a decrease in virion production and a further decrease in the infectivity of ORF36 mutant viruses. So I want to leave you with some overarching conclusions. So we study KSHV. It's a human herpes virus, and it's the cause of 3 human cancers. My work has mostly been related to 2 KSHV tegument proteins, ORF45 and 36, both of which have vital roles in the production of infectious progeny. And I guess so the goal of anyone working on human disease is to cure the disease. So we hope that by understanding the functions of KSHV tegument proteins and also delineating the mechanisms by which cellular pathways are modulated, that this may allow for the discovery of novel targets in order to treat or prevent KSHV-associated diseases. So I got a lot of requests to bring back the rap that I did at Colloquium last fall. So I'm going to leave you with that, and I hope that you enjoy it. This is my dissertation rap.
Viruses are obligate intracellular parasites and after establishing infection, herpes is kept alive. KSHVs and 2 cause malignancies. The one we study is called KSHV. It encodes almost 100 latent and lytic genes. Functions of ORF45 were a mystery before we discovered it enhances risk activity. So we wanted to address substrate specificity. In KSHV infected cells, we use lytic replication to stop decreased phosphorylation of ORF45 mutation after immunoaffinity purification and mass spectrometry. Our understanding of risk substrates improved astronomically. At the top, you see EIF4B, a substrate of risk we reported previously. We confirmed this phosphorylation with a specific antibody, and after validation proceeded immediately to knock out risk using CRISPR-Cas9, ran a Western, one of my favorite pastimes. Apparently knocking out risk compared to the virus compared to the wild type. This is the black line. A CRISPR reconfirmed what we were expecting. Knocking out the genes we selected significantly affected viral titers shown here on some future directions. But that's just one section. Now on to the next one. Another ORF, this one is 36, the viral protein kinase and a substrate of risk. I was hoping to discover a new function and did. A stable interaction. Look at that mobility shift. We mapped the binding domains using truncations, and the dephosphorylation reduced the association. Binding seems to induce potential stabilization, so at this point I'm thinking, okay, I guess I'll make some mutations. Now starting KD and back 16 OKC saw decreased expression of folate genes. Regarding the interaction between 45 and 36, a branch stability assay can experiment out on this. And finally, those mutants I was just discussing are significantly deficient in virion production. On top of that, GFP intensity is lessened. Regarding the role of 36 in primary infection, I'm so closer, and an approval for my committee. I'm getting my
So yeah, that's all I have. So, and I wanted to thank everyone who made this possible. There's a lot of people left out here, but Dr. Zhu for his guidance and teaching me how to think about science and how to design experiments. Previous postdocs in the lab, Zach, current graduate student for mutagenesis of the back. And I've been really lucky to have smart and hardworking undergraduate mentees who've made a lot of this work possible in the time that it has happened. They turned what would have been 11-hour days into 9-hour days, so I'm grateful for that. Joseph, who's here, and Brittany, who's teaching right now, couldn't make it for making one of these ORF45 mutants I used. Members of my PhD committee for their support and guidance along the way. Dr. Veer for helping with the RNA-seq analysis. Without him, I really wouldn't have been able to present anything on that. And obviously the funding sources, Ruth for help with the facts, and also confocal, even though I didn't use that data. Everyone in administrative support, all of my friends and colleagues. If I forgot anyone, I'm sorry, but all of you as well. Everyone watching on the live stream, so I know at least my brother, my girlfriend, Sarah, future wife, and a lot of other people. Hopefully they're still watching. So yeah, I'll take any questions. Thank you for your time. Yes. Yeah.
So I guess a few questions, but I'm going to focus on that one. It's easier to remember. So 45 recruits risk then phosphorylates 36. That's the idea, right? 45 looks like it acts as the scaffold.
That's what we think.
So
Yes.
You have 36 phosphorylate. I saw that you have a phosphorylation site identified. 36.
So we did, and we actually did some.
I didn't see the data that for example, you make a phospholipid now and binds out to 45.
Right. So we did do some experiments. We did try to at least make the phosphodeficient mutant because there's only one. It made it easy because there's only one of those motifs in ORF36. So when we did that and we did the co-IP, then we didn't detect that phosphorylation. So we didn't find any. So I didn't try the stability assay with that one. That would have been something to try. But for all the assays I did try, we saw no effect of that phosphorylation. So right now we don't know what
sites also phosphorylated.
it could be. Yeah, it could be bulk phosphorylation. But for that one particular site, we don't know. We haven't ascribed a function to that phosphorylation.
So the other question I may have is really fascinating, the ERF4A, because this is involved in translation. So my question is, in your luciferase assay, you're looking at the product translation, right? So the G4 quadruplex sequence you introduced, does it affect the transcription as well?
So I didn't mention it, but those data that I showed are normalized to a separate luciferase construct. So that's sort of to try to normalize the effect on transcription. So I don't, definitely ORF45 does have some effect on transcription if you just were to do one luciferase with and without 45. Seems like almost any gene could be upregulated.
So in that case, maybe the platform UTR, some kind of structure involved in the ribosome scanning is defective.
Yeah, it could be. I don't know the exact mechanism, but I guess that it's because of these structured 5' UTRs that take more energy to resolve, that without 45 and EIF4B enhancing the helicase activity of 4A, that you may not.
So all the KSHV open-reading frames that are affected mostly have the G4.
So I tried to look for.
Oh, my question is, what's the specificity?
Yeah.
For F4A to specifically regulate the KSHV genes.
Right. So the only trend I've seen is that just in general, the ones with longer structured 5' UTRs are the ones that seem to be more dependent on ORF45 and EIF4B, whereas genes with very short 5' UTRs don't seem to be dependent. So there's no sequence that I've found that we can say that this sequence motif is important, but it just looks like I basically just did bioinformatic analysis just on the minimum free energy and also the length and try to find different structures. But the only thing I could find is the trend between minimum free energy and translation efficiency. And I didn't check every gene, so that probably more comprehensive work on that would be needed. For instance, the ribosome profiling. Any other questions? I was hoping that by doing the wrap at the end, if you had any questions, you would just forget them.
So where is your postdoc?
It's at Washington University in St. Louis. So I'm going to be working in the Milbrandt lab, and this is the project that sort of drew me to that lab. So I think it's what I'll start working on, but not necessarily the only thing.
It's like a month away.
Yeah.
Are you sure they're gonna give you the PhD today?
No more questions. So I guess the committee member, let's stay. So if he did have or have celebration in future.
Okay.